Well, my name is Howard Berg, and I'm a professor at Harvard, and I've been interested in the modal behavior of bacteria for a very long time. And so my lecture in two parts is on the marvels of bacterial behavior. I first want to tell you about the history of the subject, which is amusing, and some physics. And then I'll tell you about how the cells do what they do, that is, describe the molecular machinery. Now, the history begins at the very earliest days of microbiology. It goes back to Anthony van Leeuwenhoek, shown here in a mesotint made from a painting which hangs in the Boerhaave Museum in Leiden. And what van Leeuwenhoek did was he made small single lens microscopes, and he looked at everything. And he lived into his 90s, which gave him a lot of time to look. And he described what he saw in letters he sent to the Royal Society in London, written on a quarto paper which you could fold twice, seal with red wax, and you write on the back to Henry Oldenburg, Royal Society, London, put them in the post. And Oldenburg would translate snippets of these and put them in the transactions of the Royal Society, which is how the world found out about van Leeuwenhoek's work. Now, there's a wonderful book written by Clifford Dobell called Anthony van Leeuwenhoek and His Little Animals. So people talked about molecules, van Leeuwenhoek talked about animalcules. And what Clifford Dobell did was taught himself archaic Dutch and went back and translated these letters. And it's his 18th letter in August of 1676 where van Leeuwenhoek describes bacteria. And he discovered them because they were swimming through the field of view, because they were modal. And here's a picture of one of his single lens microscopes. They're rather small. You'd hold the lens right up to your eye. There are nice descriptions of this work by Brian Ford in two books. One's called The Leeuwenhoek Legacy and the other Single Lens, if you want to learn more. And the lenses were made in various ways, but for example, you could just pull a glass fiber and then break it with a flame and let the tip swell and make a sphere, and then put that little sphere in your brass or silver plate, and then you had your microscope. Now, these microscopes were really very good. One of his forebears, Robert Hooke, used compound microscopes, which weren't as good, and these have much less spherical aberration than the compound microscopes of those days, and they're almost as good as some of the best microscopes made today. But in his 18th letter, August 1676, he says, well, this was for me, among all the marvels that I've discovered in nature, the most marvelous of all, and I must say, for my part, that no more pleasant sight has ever yet come before my eyes than these many thousands of living creatures seen all alive in a little drop of water, moving among one another, each several creature having its own proper motion. Now we can skip forward 200 years. One of my heroes is a German physiologist named Theodor Engelmann, who worked in Utrecht. And I like this slide, which you won't be able to read because it's so small, but it says here that in 1867, he went to work with Donders at Utrecht, where he married his teacher's daughter and finally succeeded him to the chair of physiology in 1888. So I like to point out to students there are more than one way of getting ahead in the field of science, and marrying your professor's daughter happens to be one. Well, the story is slightly tragic, because his wife died in childbirth, in the birth of twins, and he remarried. His second wife, Emma, was a concert pianist, and they were friends of Brahms. And so his friends chided him that he would be remembered more for his friendship with Brahms than he would be for the stuff he was doing in the lab. And that's probably true, because there is the Engelmann Quartet named after this man. But he did two kinds of experiments. First, he found a very large purple sulfur bacterium now called Chromatinum okenii. He called it Bacterium photometricum. And he discovered that the cells would back up when they moved into the dark. So if you look through a microscope at these guys swimming, pass your hand between the stage and the objective so it's momentarily dark, and you continue to look, everybody's going the other way, they back up. So he called this a shock reaction. And then he showed that these bacteria would accumulate in the spots of light, not because they liked the light, but because they didn't like the dark. So they could get in, but they couldn't get out. And they also studied the responses of different bacteria to oxygen. So here's here 3 examples, A, B, C. On the left, A, they're bacteria that like oxygen. What he did was put a drop of cells on a microscope slide, cover it with a square cover glass, and then wait half hour, an hour or so, and then see what he found. And on the left, the cells swim out to the edge where there's more oxygen. On the right, they swim as far away as they can get because they don't like oxygen. And in the middle, they like some, but not too much. And he showed that this was an oxygen response by blowing hydrogen gas at that preparation in the middle, in which case the square got larger, and if you blew oxygen gas at the preparation, it got smaller. So this era in the late 19th century was one in which people first began to study the responses of bacteria to light, to chemicals, and various other things. And then we go another 100 years or so, and we've come to Julius Adler, who was a biochemist at the University of Wisconsin, who introduced E. coli as a subject of study. And this is his first micrograph of E. coli. It has 5 flagella sticking out, but they're actually much longer than appears here because it's a negatively stained preparation, and the negative stain peters out. So it's an organism that's about 2 micrometers long and 1 micrometer in diameter. It lives in your gut. So it's a friendly organism mostly. There are bad ones, but most of them are friendly. And here's Julius Adler, but not in 1969, but in 2011. He wrote a paper for the Annual Reviews of Biochemistry describing his scientific saga. Now, he modernized an assay that had first been developed by a guy named Wilhelm Pfeffer in 1884, which is called the capillary assay or the Pfeffer assay. So what you do is you take an ordinary microscope slide, put a U-shaped tube, melting point capillary tube on the slide as a spacer, cover the U-shaped end of the tube with a square cover glass, pipette your E. coli in between the cover slip and the slide, and then you insert a fine capillary tube. That's not the scale, which it contains a few millimeters of something you want to test. And what the bacteria do is respond to the gradient of chemicals that diffuse from the tip of the capillary tube, and they gather at the tip, and then they swim inside. So if you pull out the tube and wash it off and plate the contents and serial dilute, you can spread these organisms on an agar plate, nutrient agar plate, and the next day you can count colonies, so you can get a dose-response curve. So here is one of the nicest experiments that Adler did with his capillary assay. It shows the response of a mutant E. coli and a wild-type E. coli to a sugar called galactose. So on the left, on the ordinate, is the number of bugs that get into the capillary in 45 minutes or so, and on the abscissa on the bottom is the logarithm of the concentration that was in the capillary tube initially. So the result on the left is for something that's called galactose minus permease minus. That mutant can neither transport galactose into the cell or metabolize it. So it gets no benefit from it, yet clearly it likes it. And its response is, 10 times more sensitive than the wild-type cell, which can take up and metabolize galactose and grow on the substance. Now, I should point out that these responses peak and then they fall off again. They fall off again at high concentrations because either you saturate the receptor for the chemical, or the response occurs so far away from the end of the capillary that in the course of the experiment the cell never gets there. But then the question is, why why is the response of the mutant on the left 10 times more sensitive than the one on the right? Now, one usually asks our audience that, and practically no one knows, but the answer is very simple. It's that the wild-type cell can eat the gradient. So the gradient is not as steep, so the response requires a higher initial concentration of galactose, whereas the mutant doesn't eat the gradient, so the gradient remains steep, and so it can respond at a lower concentration. So the punchline at the bottom is that chemotaxis is a matter of aesthetics rather than material gain. You respond to a chemical because you like its taste. You don't care whether you can benefit by eating them or not. No, but obviously these techniques were developed for reasons of survival, which means you learn to chase things that you can eat, but you can, in the laboratory, fool the animal by using mutants that are defective, such as the one that's galactose minus and permease minus. So here are some of the historic bacteria shown on the same scale. A there is Adler's E. coli. It has a bundle of 5 or so filaments that protrude out the back when the cell is swimming smoothly. B is Engelmann's Chromatum okenii, which is enormous, has a bundle of about 40 filaments sticking out the back. And then on the bottom there is Leeuwenhoek's little eel, which is it's a ballet dancer. As shown there, it's swimming like this, and then when it wants to go the other way, it flips its bundles and backs up as if reflected in a mirror. So it's a large cell with about 20 flagellar filaments at either end. Now, some of the physics. If you're as tiny as E. coli, only a micron in size, and you're immersed in water, you can't coast. Your mass is very small, and the viscosity of water is very large. So you can compute how far the cell can coast if it puts in the clutch, and it's less than 1/10 of a diameter of a hydrogen atom. So there's no inertia in the problem whatsoever, just viscous forces. And there's a wonderful paper I'll come to later, written by Ed Purcell, called Life at Low Reynolds Number. He tried to put himself in the shoes of a bacterium, so to speak, and figure out what life is like on that scale. Now, the other problem the cell has is it's so small it's buffeted about by Brownian motion. It diffuses, it jiggles about and migrates, and you can work out the distance it goes from the elements of diffusion, which I'm not going to try to explain. But if you assume that the bacterium is a sphere roughly of radius 1 micron, so 2 microns in diameter, and you compute how long it takes to diffuse diameter, it's about 1 second. So if you're an E. coli sitting next to another E. coli, in another second you'll be in your companion's lap. But for more important, from the point of view of swimming, is that you can't swim in a straight line because there's a rotational Brownian motion, which you can also compute. And the answer is that you wandered off about 90 degrees in 10 seconds. So that means you forgot where you've been. So any measurements you made more than 10 seconds ago are no longer relevant. So you have to respond within that time, or you can't play the chemotaxis game. So here is a movie showing E. coli swimming. These cells have been labeled with a fluorescent dye. What this movie shows is that single motors can change their direction of rotation, and the filament that connects to those motors can come out of the flagellar bundle and change their conformation and go back in again. So I got into this business a long time ago, figuring out how to follow individual cells as they move about in 3 dimensions. And what we found was that cells have 2 modes. They can swim in a very smooth manner, and we call these runs, and then they change their mind, they jiggle about and try a new direction. We call these twiddles, but nobody liked that word, so they're known as tumbles now. So runs are relatively long, tumbles are relatively short. And so the cell wanders about in a random walk. Now, if you put one of Adler's capillary tubes off to the right here and ask what happens when the cell, by chance, is going up the gradient, then the tumble gets postponed and the run gets longer. So the random walk becomes biased. Now, it turns out that bacterial flagella, in fact, rotate. They're not bending machines. They don't wave. They rotate rigidly. They're driven at their base by rotary engines. And it turns out that when you're swimming smoothly, when you're running, the several flagella on the cell are rotating counterclockwise, and they form a bundle behind the cell, pushing it forward. When the cell tumbles, one or more motors changes its sense of rotation, the filament changes its shape, and the cell jiggles about, and then the filament goes back to rotating counterclockwise, and it rejoins the bundle, and you make another run. So I have a model of this made out of spiral wires, and E. coli is like this. It's got these things sticking off in various places. And if they all rotate counterclockwise, seen from your point of view, they just go around side by side, no problem. And then if one or more motors reverses, this bundle flies apart and the cell jiggles about. And then the bundle reforms with a different orientation, and the cell swims off in another run. So it's the direction of rotation of the motors which determines the mode of motion. Now, another thing about being small and wanting to measure concentrations, it's generally true you can't count molecules without taking time. It's a counting problem where the standard deviation divided by the mean is 1 over the square root of the size of the count. So you see it in any kind of scintillation counting, any kind of counting you do. So one way of looking at this is the cell is a little spherical counting device of radius A, and it's immersed in a medium of concentration C, and so the number of molecules you get for a single count is just the volume times the concentration. And that number turns out to be rather small. So to get the count up, you have to count again and again and again, but it doesn't do you any good to count right away. You have to wait for a new set of molecules to diffuse into this region of interest, the old set to diffuse out, so that you can make a statistically independent count. And you can figure out how long that takes. It's a time tau, which is a distance squared divided by the diffusion coefficient. And so in time t, you have t over tau such counts, so your count goes up by a factor of n, which is t over tau. And so when all is said and done, the change in standard deviation concentration delta C divided by C is inversely proportional to the time that you count, as you can see in the lower right of this slide. Now, since t is in the denominator, you can make that number arbitrarily small just by making t arbitrarily large, but the cell can't do that because it only has about 10 seconds before it forgets where it's been. So that puts a limit on the precision that the cell can obtain, which puts a limit on the concentration that it's able to detect. And so it's much higher, actually, than what a eukaryotic cell like a leukocyte can do, which can take a very long time. It's on the order of micromolar, the best the cell can do. And that turns out to be the association constant on the receptors. So E. coli understands that physics. Now, just to remind you how much time the cell has, you can take a tethered cell shown in the inset there that's cyan in color, and you can bring up an ionophoric pipette, and you can spritz a negatively charged attractant molecule on the cell any way you like. And in particular, if you give it a short kick, a small pulse, you get this biphasic response. So the cell, in effect, swims smoothly for about a second, and it tumbles more for about 3 seconds, and it comes back to its initial behavior. So you get a biphasic response that lasts about 4 seconds. So it's fair to say that E. coli has a short-term memory, but it's only about 4 seconds long. So what the cell does is it counts the things of interest in its environment over a second, looks up what it got for the previous 3 seconds, and then responds to the difference. Now, one final problem is that if you don't swim far enough away, you haven't gotten away from your local environment. So you have to outrun diffusion of attractants and repellents in your local environment. So to put it another way, as it says in fine print there, to find out whether life is getting better or worse, the cell must swim farther in time t than the distance over which molecules reach it by diffusion. So that says that the distance that you swim in time t, which is swimming velocity times time, has to be larger than the distance that molecules can come to you via diffusion, which is the square root of d times t. That gives you a condition on the time, which is the diffusion coefficient divided by the square of the swimming velocity. So cells typically swim 30 microns a second. The diffusion constant for a small molecule that the cell wants to eat is maybe 10 to the minus 5 centimeters squared per second. If you work that out for a swimming time of about a second, you have to move about 30 microns. So if you look back at the track of a swimming cell, the runs are on the order of 30 microns long. So, and when the cell responds to a gradient, the runs get longer. They don't get shorter because it doesn't do you any good to scrub around in the local environment. You have to go far enough away to outrun diffusion and find out whether life is really different. So E. coli generates thrust by using viscous drag. And the secret of this game is that the helical flagellar filament is a thin thing, and segments of it can be regarded as a series of little thin sticks. And if the stick moves sideways, the viscous drag on it is about twice what it is if it moves lengthwise. And if you work that out, you discover that this filament, when rotating, will generate both a thrust and a torque. So as the cell swims from left to right, as shown there, the cell body rotates back the other way. To balance the torque required to rotate the filament. And the thrust generated by the filament is balanced by the viscous drag on the head as it moves through the medium. So everything's fine. Now, if you don't believe in rotation, here is a wonderful experiment done by Mike Silverman and Mel Simon back in 1974. You just break the filaments off of a cell and you bolt the cell down by one filament, and then what happens is the cell rotates. So the motor, instead of driving the filament, drives the cell body, or as it were, the boat. To illustrate that, I brought a mouse here, which has red pants, and it has rotary arms. So if you wind him up and hold him by the tail, the arms go around. But if you hold him by the arm, then the mouse goes around. So this is a tethered mouse. It's a smooth mutant because it only rotates in one direction. But it illustrates the tethered cell technique. And the other thing you can see in this movie is that the little guys are spinning really fast, and the big guys are going around slowly. So in this domain, the motor generates constant torque. So if you increase the load on it by making the thing you want to rotate large, it slows down. Now, there's a very peculiar thing about motion in a highly viscous medium. That is that reciprocal motion does not work. And this was first recognized by a guy named Wilhelm Ludwig, who published a paper in a German journal of physiology in 1930 that nobody read. So if you read the hydrodynamicists, they say, well, Sir Geoffrey Taylor discovered all this in 1955 or whatever. But no, it's a much older discovery. But so Ludwig's had an organism that has two oars, two rigid oars that are sticking out that go up and down synchronously. So you can swim if you're large by going down fast, coasting up, and recovering slowly. So then you swim like that. Yeah. But if you're tiny, if you're microscopic in size, you go up fast and you go back slowly, but you don't go anywhere. You can't swim. Now, this idea got immortalized in a wonderful paper by Ed Purcell, a Harvard physicist, called Life at Low Reynolds Number, published in January 1977, I think, where the simplest organism he could think of that would suffered from this problem would be a scallop, shown in the lower left there. Now the scallop just has one hinge so it can open its mouth, close its mouth. And that's of necessity a reciprocal motion. So if you're a macroscopic scallop, Purcell argued, you can close your mouth fast and coast, open it slowly, fast and coast. But if you're microscopic, you open it and you move below it, close it, you move below it, and you just go back. You don't go anywhere. Now, after he published this paper, there's a guy named Charles McCutcheon wrote him a letter at NIH. He said, well, that's all very nice, but I have a tank full of scallops in my office, and they don't swim this way. They swim this way. They go exactly the opposite direction that Purcell said they ought to be swimming. And I like this because it means if you're a physicist thinking about biology, you get in trouble because biology is always more complicated than you might imagine. So what the scallop actually does is blow water out the sides of its mouth. So when it closes its mouth, it actually goes that way. It's hard to do this because it seems so unnatural. This is easy to think of, but to go that way takes a little practice. Okay, now I have a demonstration of this. The fact that reciprocal motion won't work. It's a beaker with a cylinder in it that you can rotate, and there's a gap between the cylinder and the beaker that you can put in a bit of dyed. We use glycerol because the Reynolds number, so-called, is small at low Reynolds number, life at low Reynolds number, and the Reynolds number is a length L, a velocity v, density rho, and a viscosity eta. And so if you have a macroscopic model, the distances are large, and the velocity is large, and the density of the fluid is just the same. But so you need to use a fluid that's quite viscous to get that ratio to be small. So in this experiment, we use glycerin, which has a viscosity about 1,400 times that of water. So what we're going to do is put a stripe of colored glycerin in the gap between the inner cylinder and the outer beaker. Whoop. Disaster. One of the risks of the experiment is you end up being blue. Okay, you can see the stripe, and it wasn't meant to be at the top there, and that won't work very well. But what we're going to do is mix this up. So you can see the dye coming around. It's funny, it's kind of slanted. Now, I can stop and think and talk and do whatever I want. I don't have to do this continuously. I just have to do it sufficiently slowly that the Reynolds number remains small. So now it's reasonably well mixed. Now we'll unmix it. We go back the other way. Again, it doesn't have to be continuous. It just has to be reasonably slow. Now you can see it's beginning to clear. There we are, look at that. So this is life at low Reynolds number, and this is a very dramatic demonstration that reciprocal motion doesn't work, and E. coli is figured out by using helical filaments to beat this problem and live a rich and long life. So in summary, I've tried to tell you a little bit about the history of this subject, which is colorful, and about some of the physics that E. coli has had to learn in order to survive in the face of very severe constraints, to be able to swim at very low Reynolds number, to be able to count molecules in its environment and decide when all is said and done whether life is getting better or worse.